Okay, now we're going to move on to the second half of Chapter 2, which is the biochemistry portion, the chemistry of living matter. Just like with the previous chapters, you will have a list of objectives that you should be able to define and go back and answer after you've listened to this lecture and read the material from the book. Alright, there are basically two types of compounds. There's organic and inorganic. Organic compounds are anything that contains carbon. They are bonded with covalent bonds, which remember that means that those are strongest bonds, and they're large molecules, which we call macro. Macro means big, so these are macromolecules. Your inorganic compounds are everything else, everything that's not bound with carbon. They use ionic bonds. Ionic bonds, remember, dissociate in water. So these are going to be smaller molecules like water and other ions. Now, let's talk about some of the inorganics like water. Why is water important? Well, it has a high heat of vaporization, which means it goes from a liquid to gas, requiring lots of heat, and results in the loss of large amounts of heat. Second, it's a universal solvent, which means that it's going to be dissolving ions for us, acting as hydration layers, and it acts as a transport medium. Most of your blood is water, and so it's transporting different ions and molecules from one place of the body to another. The third thing is cushioning. It will actually make little water beds to protect organs, like around your kidneys. Some other important inorganics are your salts. Salt, everybody thinks well, I put salt on my food, but it's a different kind of salt per se in your body. Now, for example, salts again are held together with ionic bonds, which means they will dissociate when placed in water, or break apart. For example, salt, sodium chloride. When we put this in water, it breaks apart into the individual sodium and chloride ions. Now refer back to the last chapter we just did, which was the first part of Chapter 2. And you remember when we looked at the bonds, when sodium donated an electron, that made it less negative or positive, which we called it the cation. Where chloride accepted that electron, making them more negative, making them the anion. Now, electrolytes are salts. They conduct electricity. So there's lots of electrolytes in your body, and we all kind of have a clue what electrolytes are. We know that they're important because remember all the Gatorade commercials, they tell you got to have your electrolytes. It's super important, and as you work through this course, you're going to find out why it's so important. Every nerve impulse, every muscle contraction, every brain wave, all these require certain balances of certain electrolytes. We'll get into that later. Now, what are some other vital roles in the body for salts? Well, you need calcium for your bones. Iron you need to carry oxygen and hemoglobin. There are so many important uses for salts in the body. Alright, in this illustration you can see that we put a salt block into a beaker of water and it dissociates. It breaks down into the individual sodium and chloride ions. Alright, some other inorganics, we've got acids and bases. Acids release hydrogen and are therefore proton donors. So we take HCl, which is hydrochloric acid, it's found in your stomach. It breaks down into free hydrogen and chloride. Bases release OH, which is hydroxide, and are proton acceptors. So we take NaOH, which is sodium hydroxide, and it breaks down into sodium in the individual hydroxide. Now, pH. You can't talk about acids and bases without talking about pH. pH is a measure of the amount of hydrogen. Now, acids and bases can neutralize each other. For example, you take that hydrogen and a hydroxide together, they make water, which is perfectly good for your body. Whereas all these free hydrogens or all these free hydroxides could be potentially very bad. Now, your body uses buffers. Buffers resist abrupt or quick pH changes in bodily fluids, and they're typically weak acids and bases. Alright, here is your pH scale. It runs from 0 to 14, with 7 being neutral. Acids go from 0 to 6.99. Bases go from 7.01 to 14. The closer to 7, or the closer to neutral it is, the weaker it is. So, for example, an acid at about a 6.5 is close to 7, so it would be a weak acid. Where an acid around a 1 or 2 is far away from neutral, so it would be a strong acid. Same thing works for the bases. If you have a base around an 8, that's going to be a weak base because it's close to 7, whereas a strong base would be around a 13 or 14. Alright, so let's do a little sample question. A solution with a pH of a 6.4 would be considered a what? Okay, well remember, first you decide if it's an acid or a base. So if it's below 7, it's an acid. If it's above 7, it's a base. So 6.4 is definitely an acid. Now remember, the closer it is to 7, which is neutral, the weaker it is. 6.4 is pretty close, so I would say that that is C, a weak acid. Okay, now let's move into our organic compounds. Again, this is anything dealing with carbon, and they are held together with very strong covalent bonds. These are your macromolecules, your big. Now, we use that term macromolecules throughout the semester, and it seems like students don't really learn the term, so it kind of confuses them later on. All right, you got to get this down. There are just 4. That's it, just 4 macromolecules. Carbohydrates, lipids, proteins, and nucleic acids. And again, you may remember that these are important molecules, but you forget that they are macromolecules. So try to keep that in. Now, carbohydrates are your primary fuel source. This is the energy, the free, quick-using energy in your body. It's so important. Lipids, these are fuel storage, and they are long-term energy reserves, but they have other uses in the body as well that we will get to. Proteins, oh my gosh, they're so important. They have structural and functional properties like enzymes, and we're going to get to all that in a minute. And then, of course, your nucleic acids, which will store, retrieve, and transmit information. Okay, we're going to start with carbohydrates. This is your first macromolecule. They're made of carbon, hydrogen, and oxygen. Now, a monomer, mono meaning one, that is the simplest form of a carbohydrate. So this is what we call our basic building block. Okay, so for carbohydrates, that would be a monosaccharide, which is a simple sugar. It's one sugar. Now, rarely do we deal with just monomers, especially if we're eating. We never eat a monomer. We eat polymers. Polymers are many blocks. So this would be poly, meaning many. You have a polysaccharide. This would be a complex sugar. Now, for function, again, the biggest thing with them is they are our primary fuel source. Okay, now they do have structural. We have cellulose and chitin. But again, nutritionally is our energy. We look at starch and glycogen, which we're going to talk so much about glycogen this semester. You'll know it like the back of your hand. Alright, so let's look and see what these sugars look like. In the top picture, these are all monosaccharides. This is one sugar. So glucose is by far the one we're going to talk the most about. But you also have fructose, galactose, deoxyribose, and ribose. Now look at the picture at the bottom. This is a polysaccharide, so meaning many. This is several sugars put together. And this is a picture of glycogen, which is a complex sugar used frequently in your body. Alright, let's move on to the lipids. Lipids are carbon, hydrogen, and oxygen. This time they have longer chains of carbon and hydrogen. That makes them different from lipids. Now, the monomer here is a fatty acid and glycerol. Now, remember, monomer means one. So a polymer would be multiple ones. These would be your triglycerides, phospholipids, and steroids. Now we're going to be talking a little bit more about those in a second. Some of you might be familiar with the word triglyceride. Maybe you yourself have had some blood work done, or mom and dad, grandma and grandpa, and they probably talked to you about triglycerides. This is how much fat is floating around in your bloodstream. This leads to your cholesterol levels, and we like to keep your triglycerides low. Now, why? Well, you don't want fat floating around your bloodstream, right? When you get to the heart chapters and the blood vessel chapters, we'll talk about more about how, deadly high triglycerides can actually be. And then we'll talk more about phospholipids and steroids in the next couple of slides. Now for your functions, again, we can store energy, we can insulate, and we can protect. Okay, lipids are essentially fats. Okay, so look at insulation. We have in the winter, typically people will put on a little more winter weight, and that's to actually insulate those core organs, like, the hearts and the stomach and liver and all that good stuff. Now, we don't need a lot because, let's be real, we live in a civilized society where we have heating and air conditioning and all that. So some of us may put a little too much insulation on. Now, what about protection? Well, you have a fat layer around your kidney, and you think, oh, there's fat around the kidney, that's bad. No, it's perfect, because the kidneys sit right against your back. We hit our back all the time. We get kicked in the back with sports and fall, and without that fat layer, the kidney would have to take the brunt of that hit, which would then damage it, and you'd be in dialysis. So fat is good around organs, depending on the organ and depending on the severity. All right, let's look at this. This is a illustration of a phospholipid, which is a major component of the cell membrane. So if you look at the picture on the bottom, you can see all the carbons and hydrogens and oxygens. And then to the right of that picture, that is what the phospholipid looks like. Okay, you have the polar head, the nonpolar tail. And think back to the first part of this chapter 2, the basic chemistry, where we talked about polar. Polar, remember, meant an uneven sharing of electrons. So it's going to give a charge, where nonpolar had an equal sharing of electrons, so there was no charge. Now, your entire cell membrane, and membrane is the outer layer of the cell, the whole thing is made up of these phospholipids, which is super important. So one of the things that you're going to get into later on in this course is, what do we eat and what is it used for in our body? Everybody knows fats can be used as energy, but they forget that before they're ever used for energy, they have to be used to replace things in your body. Okay, prime example, you have skin. We slough off skin every single day. Okay, well, we have to make new cells. Or eventually we're going to be see-through, right? Yeah, that's not going to work. So we need new skin cells. Well, to make a new cell, you have to have thousands of new phospholipids. So part of that fat you're eating today is going to be making you new skin cells and, whatever other cells that you need in your body. Steroids. Steroid is a type of lipid. All right, so there's two ways here. First, we've got the cholesterol. Now, cholesterol is a membrane component used for stability. So when we hear about cholesterol, heck, I just mentioned cholesterol. We say, oh, cholesterol is bad. Okay, different types of cholesterol. The cholesterol that we're referring to here is actually adds stability to the cell membrane. Now, you also have, of course, cholesterol made by the liver, which is still used good. The problem with cholesterol is when we eat too much cholesterol. Your body makes some, and you don't need as much. But we'll get to that later on. Now, what else is steroids used for? Steroid hormones. That's a huge thing. What's in a steroid hormone? That's like estrogen and testosterone, which are super important hormones in our body. So again, yes, we can use fats for energy, but first we've got to make our estrogen, our testosterone, use the cholesterol for the membranes, phospholipid. We got to do all this stuff before we can use it for energy. All right, let's move on to proteins. Ooh, I love some proteins. Proteins are made from carbon, hydrogen, oxygen, and nitrogen. Our monomer here is an amino acid, and there are 20 different kinds. Now, a polymer is going to be a polypeptide. Okay, what's a peptide then? A peptide is a string of amino acids. So think of amino acids like little linking logs. Individually, they're just a linking log. You start putting the linking logs together, you make up polypeptides, which are complex proteins. And they have so many functions in the body, from cellular structures to cell identity. When you get into the cell chapter, you'll be talking about how cells communicate with each other. How do they know that a liver cell is talking to a kidney cell? Well, there's a protein on the surface of the cell that tells it that. So, how does the sperm know to find the egg? Same type of thing. Now, proteins have a very specific structure, and there are 4 different types of structure. We start off with the simplest form, which is a primary structure. This is simply a string of amino acids. Very simple. Then you move on to a little bit more complex with secondary. This is amino acids strung together in an alpha helix or even a beta pleated sheet. Tertiary, even more complex. Now the proteins are being folded based on their bonds. Quaternary is the most complex protein. This is 2 or more polypeptide chains. They have to be folded properly. This gets very complex very quickly, and all kind of things can go wrong. What if the protein was folded the wrong way? In a second, we're going to talk about how important it is for a protein's function is directly related to their structure. So if you change the structure of a protein, you've now changed the function of that protein, which can cause havoc through the body. Alright, so here on the left you can see a primary protein structure, which is just a string of amino acids. To the right, you see the alpha helix and the beta pleated sheet. Now we're increasing in complexity. On the left, you can see the tertiary structure. Again, we're starting to fold these proteins. And to the right is quaternary. That just looks crazy complicated, doesn't it? An example of a quaternary or a very complex protein in your body would be hemoglobin. Hemoglobin is found in every single red blood cell, and it carries oxygen for us. All the oxygen in our body is carried by hemoglobin. Well, most of it at least. And this is such an important molecule in our body. Alright, denaturation. That's when proteins fall apart and stop working. Okay, so something disrupts the bonds, which then disrupts the shape. If you alter the shape, you alter the function. That's super important. And right now I know this is also very tiny, and how do you fit all the pieces together, and when is this going to matter later on? Well, it's going to matter hugely later on, because you're going to talk about different things like imbalances in the body. Oh, here's a great example. Alright, enzymes, you're going to learn in just a second, are proteins. Proteins are used for lots of stuff. What do we use enzymes for? Enzymes speed up reactions, but they only work in certain environments. And if you change that environment, you denature it, and then it doesn't work anymore. What about hemoglobin? What if you were folding hemoglobin wrong? Well, now you've altered the function. It won't carry oxygen, or maybe it won't carry as much oxygen, but it's definitely not working the way it should. So there's all kind of havoc that can be, wreaked because of this, but you'll get into it later on. Now, how do we disrupt the bonds? Well, pH is an example. Most proteins work within a very narrow pH. If you go out of that range, then they fall apart. Temperature, same thing. One of the things we worry about with high fevers is that proteins denature and stop working. So hemoglobin stops carrying oxygen for you. If vital proteins and enzymes in the brain stop working. What else disrupts a bond? Ionic concentrations. Certain ions around them will then interact with them. So if you change the ions, they don't work as well, or maybe not at all. Same thing with mechanical agitation. That's shaking. Okay, one of the things that we talk later on about is like when you draw blood. Okay, the blood goes in the test tube and you gently roll it. If the person taking the blood shakes the blood, well, then the hemoglobin inside the blood will rupture, and all of a sudden, you'll go to analyze that blood and you'll be like, oh my God, there's hemoglobin everywhere, this person's dying. No, you shook the blood, you denatured the protein, and all went to heck from there. Enzymes. Oh Lord, we're going to talk about some enzymes in this class. Enzymes are just proteins. Now, most enzymes, their names will end in ASE, like amylase and peptidase and all these good ones. Now, not all, but most. They are biological catalysts, which means that they speed up chemical reactions. And they do so by lowering the activation energy of a reaction. Okay, what does that mean? Well, let's look at our slide. Okay, here is a picture talking about enzymes and activation energies. If you look at the first or top picture, Notice that this little red ball has got to go up. That's all the energy it's required to start that reaction. If you look at the bottom, we've added an enzyme now. It doesn't require as much energy. So now the reaction can start much sooner because you're not having to wait to build up those energy. So that's how enzymes speed up reactions, by lowering the activation energy, the energy it takes to start the reaction. Now, enzymes fit together like puzzles. So, enzymes and substrate. What's a substrate? A substrate is a generic term that means anything being chemically broken down. So, enzymes and substrate fit together like pieces of the puzzle. That's how that enzyme knows that it's supposed to break down that substrate, because they fit together. If they don't, then it's not supposed to work on that, and it'll move on. Okay, now we're at our last macromolecule, which is a nucleic acid. These are made of carbon, hydrogen, oxygen, nitrogen, and phosphate. Our monomer here are nucleotides, which are pentose sugars, phosphates, and nitrogenous bases. A polymer would be several of these put together. That is what is our DNA and our RNA. What does it do? Well, it stores, it retrieves, and it transmits all of our heredity information. So let's look at one of those polymers, DNA, which stands for Deoxyribonucleic Acid. This is a double-stranded helix in the nucleus of the cell. Now, DNA is trapped inside that nucleus. It doesn't get to come out. That's going to cause a little problems for us later, but we'll deal with that when we hit the cell. Now, your nitrogenous or nitrogen bases, there are 4 of them: adenine, guanine, cytosine, and thymine. Again, when we get into the cell chapter, we're going to be talking about DNA replication, or making copies of DNA. These bases have to be paired with each other, and there's highly specific. But you'll get onto that later. Just foreshadowing. All right, what's the function of DNA? Well, again, it has to make a copy of itself for all your new cells. Like what we were talking about with our skin sloughing off. Every new skin cell you make has to have a copy of the DNA in it. So, you're constantly making copies of DNA. It also provides a genetic code for making proteins. Every protein in your body is made based on a recipe from DNA. But again, you'll get into that in a little bit. Another polymer of nucleic acids is RNA, which stands for ribonucleic acid. This is single-stranded. It's found in the nucleus and in the cytoplasm. So this is going to work out good for us because it isn't, or it is not, trapped in the nucleus. It can leave and come out into the cytoplasm of the cell. That will play a vital role in protein synthesis, which is the making of proteins in the body. Now, the nitrogen bases here are adenine, guanine, cytosine, and uracil. Notice that 3 of them are the same as DNA. The different one is DNA had thymine, where RNA has uracil. So notice that change. Now, there are 3 types that function in protein synthesis. There is messenger RNA, transfer RNA, and ribosomal RNA. And again, you will be dealing more with these when you go over the cell chapter. Adenosine triphosphate. This is ATP, probably the most talked about thing in any anatomy and physiology course. This is the source of immediate usable energy. And oh my gosh, everything in your body pretty much requires ATP. There's a few things that don't, but most do. And so you'll be hammered with ATP in this course. Now, what is it? It's an RNA nucleotide with an adenine and 3 phosphates attached. Thus its name, adenosine triphosphate. Again, we use ATP for so many things. For example, if you look over at the right, the top picture is showing membrane transport, moving stuff in and out of the cell. Second picture is talking about the contraction of smooth muscle. Okay, but we have so many others. So skeletal muscle, cardiac muscle, it all still requires ATP. And then chemical work, simply for reactions to take place, requires energy. Alright, so let's give a little recap here, give you a couple of quick questions, see how you got this last bit of material. The best buffers are going to be A, strong acids and weak bases, or B, strong acids and strong bases, C, weak acids and weak bases, or D, weak acids and strong bases. So go back and look and where we talked about pH and acids and bases, and it specifically says what a buffer is and which one works best in the body. The answer would be C. Weak acids and bases are the best buffers in the body. Now, let's do a second question. While doing some biological research, I came across an unknown molecule. I determined that it contains glycerol and long chains of carbon and hydrogen. This molecule is most likely a what? A, carbohydrate, B, lipid, C, protein, or D, nucleic acid. Go back to those macromolecules and see which one of them had glycerol. Okay, give yourself a second. You should see that it is B, a lipid. Alright, you did good. Go back, look at your objectives, make sure you can answer those, and then feel free to move on from here.